Image This is Sharon Stack, BNF Dunne and Elizabeth Riley, Director of Notre Dame's Harper Cancer Research Institute. View more Credit Matt Casher 2 papers involving ovarian cancer research at the University of Notre Dame's Harper Cancer Research Institute HCRI, one featuring new research and the other a review article, were published as cover stories in their respective journals. Sharon Stack, BNF Dunne and Elizabeth Riley, Director of HCRI, along with postdoctoral researchers Jing Yang and Yulia Klemenko, among others, have been studying why ovarian cancer cells uniquely spread throughout the peritoneal cavity. Within that abdominal space, shed ovarian cancer cells bind together, forming lethal structures called aggregates. Those aggregates float in the cavity until eventually sticking and penetrating more tissues. This mode of metastasis, along with vague symptoms that cause diagnostic delays, makes ovarian cancer one of the most deadly cancers. In their article, published in the Journal of Biological Chemistry JBC, Yang, Stack, and colleagues studied a membrane protease called MT1-MMP, which is an enzyme that begins the breakdown of connective tissue proteins. MT1-MMP is generally not detected in normal ovarian tissue, or even in benign ovarian tumors. MT1-MMP includes a short cytoplasmic tail, or chain of 20 amino acids, that regulates activity on the cell surface. The tail contains 3 potential places where proteins are regulated, called phosphorylation sites. Yang's and Stack's research demonstrated that phosphorylation may play a role in regulating the transition of ovarian cancer cells from the floating phase to the sticking phase in the peritoneal cavity. MT1 is a molecular clipper on the surface of cells, clipping the proteins out of the way just like we would clip away roots of a tree, to allow more room for other plants, Stack said. For ovarian cancer cells, this makes them stick better and gives them room to thrive. And becoming stickier means more metastatic success, effectively spreading the cancer to new areas of the body. The discovery is important because understanding how the different phosphorylation sites function could lead to drug discoveries that target and inhibit the process, Stack said. The review paper, published in Cancers in August, is a 32-page overview by Klemenko, Stack, and Oleg Kim, an adjunct research professor in the Department of Applied Computational Mathematics and Statistics, that summarizes current research into ovarian cancer metastasis and chemoresistance. The paper also outlines computational models that could help understand these processes. Stack said she and her team were honored to have their work published on the covers of 2 journals this summer, the 3rd time her work has been featured on a cover, and the 1st time to have 2 articles on covers in 1 month. Another study evaluating the effect of parity, how many times a female has delivered babies on metastasis, has been accepted in Cancer Letters. A study about aging on metastasis has been submitted and is undergoing review. The research we're doing at Harper is leading us closer to answering questions unique to ovarian cancer, Stack said. This will eventually pave the way for more and better treatment options, with the goal of increasing the survival rate for women with ovarian cancer. The JBC study is the 3rd in the summer's list of published research into ovarian cancer metastasis that originated at HCRI. The other studies were published in Neoplasia and Oncogene. The most recent study was funded through the National Institutes of Health and through the Leo and Ann Albert Charitable Trust. Disclaimer: Oz and EurekAlert are not responsible for the accuracy of news releases posted to EurekAlert by contributing institutions, or for the use of any information through the EurekAlert system.